Hi, everyone. My name is Tyson Dawson. I'm a PhD student at the George Washington University in Washington, D.C. Today, I want to show you the results from a study we are currently wrapping up in which we take a three-pronged approach to the analysis of metatranscriptomic data from nasal swabs of COVID-19 positive patients. These samples were collected early on in the pandemic in the state of New Jersey, and they have rich metadata, which allows us to study the role that comorbidities, patient medications, clinical measurements, and disease outcome have in the context of viromics, metagenomics, and host transcriptomics. So we look first at the viral genome of each sample, second at the bacterial abundance and diversity in each sample, and then finally the host transcriptional response between patients. Briefly, I'll go over the workflow that was conducted for this study. As I mentioned, our samples were collected from the Hackensack Meridian Health Network in the state of New Jersey, and the east coast of New Jersey is part of the New York City metropolitan area. And the analysis of samples from this region gives us a window into the early events of the pandemic in this region, which was hit especially hard in mid-2020 from about March to July of 2020. The patients studied were primarily white or Hispanic, and the majority of the patients were over the age of 60. Samples were collected via nasal swabs, and then targeted sequencing was performed for viral genomes, and metatranscriptomics via RNA-seq was conducted to capture bacterial and host reads, which were then mapped using Pathoscope, which uses a Bayesian reassignment framework to handle ambiguous reads. Multiple sequence alignments were created and analyzed for the viral genomes. Microbial abundances across samples were measured. Phylogenetic analyses were performed, and then clustering revealed groups of samples, and host transcriptomics also revealed differentially expressed genes and pathways. So this is just kind of an overview into the study. And so let's kind of jump into the first prong that I mentioned, which is the viral genomes. Up here at the top, you have a phylogram, which is annotated with different metadata. Most of our genomes were from the B lineage. This is defined by the Pango nomenclature. Only one, this one here in orange, was from the more ancestral sort of Wuhan lineage. And as we expect, we didn't see very many significant really any significant correlations with patient attributes like sex, race, age, etc. Down here at the bottom, we also identified mutations which were common in our samples. This is shown in this lollipop plot here. Lollipops with a red dot at the end mark missense mutations and are annotated with corresponding amino acid substitutions. The height of the lollipop indicates the frequency, and the x-axis is genomic position. In this time period in New Jersey, we already see that the D614G mutation here is very frequent, and it soon became just about omnipresent in all samples today. We also observed this Q57H mutation, which is associated with increased transmissibility but lower mortality. And we also see this NSP12P23L mutation, which seems to have co-evolved around the world with the D614G mutation. Next, we wanted to see whether variation in gene regions was significantly associated with our metadata of interest. For each of these gene regions, we composed a distance matrix in which the genetic distance between each pair of samples for that region was calculated. And then we used those distance matrices to form clusters. And then for each metadata variable, we tested whether the structure of the clusters could be explained by the metadata, and we calculated a normalized mutual information score and a p-value from a Permanova test. And so in this heat map, cells which have a dark color and multiple stars, like this one here, are important hits from this test. The variation in the NSP10 region, for example, seems to explain the amount of time a patient spent on a ventilator. This gene region, NSP10, is involved in helping the virus mimic cellular mRNAs and prevent host recognition. Similarly, the structure of NSP4 over here seems to explain levels of C-reactive protein, which is an important marker of disease severity. This general approach highlights how mutations can create variation in gene regions, which subsequently have biological consequences that can change disease outcome. In our last step of this viral reads analysis, we wanted to determine if any of our metadata were significantly associated with viral load. So to determine viral load, we aligned our metatranscriptomic reads to a database with the RefSeq genomes of all viruses. And then we extracted the number of reads from each sample that aligned to the SARS-CoV-2 genome. Then we log transformed these counts and performed ANOVA tests for categorical variables and correlation tests for numerical variables. And we showed that 8 metadata, which are shown here, are significantly associated with this viral count value, which is sort of a surrogate for viral load. These 2 in the bottom left are basically positive controls because our metadata came with cycle quantification values for 2 SARS-CoV-2 gene regions. So we would expect these to be correlated in this way. And interestingly, we show that patients with cancer seem to have higher viral load, and also patients who died from COVID-19 had higher viral loads than those who did not. Next, we wanted to identify those bacteria which were significantly different between conditions of interest. Again, this is based on our metadata, and we use Maslin-2 to test for that significance. And in this heat map, we highlight the subset of bacteria that contains the top 50 most significant associations. Corynebacterium tuberculostericum, for example, is significantly more abundant in patients who have a cough and also have long hospital stays or long stays in the ICU. It's a multi-resistant organism that has been previously identified in patients who had spent a long time in the hospital, so makes sense. Very similar, we see Haemophilus hemolyticus here, a pathogen with similar characteristics that has also been observed, that in this study was observed in greater abundance in those patients who spent longer periods of time on a ventilator. The question still remains whether the abundance of these bacteria is causative or actually just a downstream effect of infection, and that's still in doubt. So we wanted to continue to probe the properties of the bacterial communities in our data. To do that, we did some hierarchical clustering at the genus level, which revealed the emergence of 2 groups of patients that seemed to be well explained by alpha diversity as calculated by the Shannon index, shown in this sort of green shaded region here. And then we further characterized the relationship between alpha diversity and patient metadata and identified that alpha diversity was significantly lower in patients with shortness of breath, fever, patients who are administered either ceftriaxone or acetaminophen, and that creatinine levels and BNP levels were similarly positive associated with alpha diversity. Still, the question sort of remained whether the abundance of opportunistic pathogens was a consequence of extended hospital stays and general host weakness brought on by disease, or whether a loss of richness and evenness in the bacterial community led to this sort of more severe disease outcome. So in order to test this, we employed a unique mapping strategy, this bacterial gene family analysis, in which we aligned our bacterial reads not to the genomes of interest by taxa, but instead to gene families. And then we conduct gene set enrichment analysis to reveal the dysregulated biological pathways, not in the host, but in the nasal microbiome, so among the bacteria. And the microbes in the nose are known to play a key role in mediating host immunity. And we see that in those patients with poor disease outcomes like high neutrophil count or a long duration on a ventilator tend to have bacterial communities which are not as involved in immune system processes, which is shown by this sort of blue hue across all of these processes. So although the link is still not robustly established, we propose that a loss in bacterial gene function in the nasal microbiome may play a role in the progression of severe COVID-19. Finally, we used the reads which mapped to the human genome. So these are host reads to probe the gene expression patterns across our cohort. We conducted differential gene expression analysis followed by pathway enrichment. And we highlight, for example, that cilium movement and assembly are significantly upregulated in patients with a fever and patients who are ultimately discharged from the hospital. Also in patients who had high levels of bilirubin and elevated LFTs. Replication of the virus in the nose leads to a loss of the ciliary layer, and these are these cells that beat rhythmically in a coordinated fashion and move mucus towards the laryngopharynx, and that helps prevent the accumulation of particles in the lungs. So this is kind of an interesting biological process that seems to, in those patients who fare better, these gene pathways tend to turn on. And that's it for my presentation. I'll just go to this last slide and thank my fellow lab members as well as the collaborators that I had for this study and the funding that we received. And thank you very much.